Okay, so all of you that are here, okay. So all three of what, four of you can actually see the text. So you see it moving.
Yes.
Okay. All right. So what I'm going to do is I'm recording this right now. So I'm going to go ahead and go through chapter 3, all right, and talk about each of the sections. Um, Try and follow along. It's going to be kind of fast, but you'll have it recorded, and I'll end up putting it online so that you can go back to and refer to it. If you come up with some questions, just you can stop me unless it's really, a lengthy question. Then just wait till afterwards. Okay. So let's go ahead. Let me make sure it's recording. Yeah, it's recording. Okay. So this chapter is chapter 3, and it's cells. Okay. So cells are the smallest unit of life. And you can see I'm starting right there at the introduction. You'll see that we have a spotlight right below that has a picture of a cell. Okay, so cells are the smallest unit of life, and we use microscopes to look at them. We cannot see them typically with our unaided eye. So this is called microscopy, right? So this is the process of using a microscope. In our school here, we typically use a 400 times microscope, but we can use 1,000 times. We have 2 microscopes down there that will do 1,000 times. But we typically use just a light microscope. Okay. So what are cells? So cell theory in its current form, we can summarize the basic concepts as cells are the building blocks of all organisms. All cells come from the division of pre-existing cells, so they replicate or they reproduce. Cells are the smallest unit that carry out life, so all of our trillions of cells that we have in our body are actually alive. And each cell maintains homeostasis, and homeostasis is defined as dynamic equilibrium. All right, so homeostasis, all cells must go through this homeostasis. Okay, so that is where cell maintains its internal environment. Okay, so the study of cells is called cytology. So anytime you see cyte, it refers to a cell, and of course anything that's an ology is the study of. Okay, so cytology is part of the broader discipline called cellular biology, which integrates aspects of biology, chemistry, and physics all in combined. So we have human body contains 2 general classes of cells, the sex cells and somatic cells. And somatic just means body or soma. If you look here, it says soma cells is body. So somatic cells are body cells, and sex cells, these are germinative cells or reproductive cells. So they're either the sperm or the oocytes. Now if we get to the reproductive system, that's when we'll talk about those. For now, we're just going to concern ourselves with somatic cells or body cells. Okay. All right. So anatomy of a model of a cell. Now what I'm going to do is I'm going to jump into the spotlight here. So this is a really important page, this picture here. So this is the spotlight of the cell. Typically in class, I would have you guys actually draw a cell, and then you would go through and add in each and all of the organelles, which just means little organs. So the organelles, and they have a specific function. Okay. All right. So let's go through and talk about the cell. Now imagine this cell is a dodgeball, okay? A circular thing, this whole thing. The rubber on the outside would be what we call the plasma membrane, okay? Or the cell membrane, so the plasma. So it's not a cell wall. The only place we find cell walls is in plant matter, right? Okay, so as we look here, you have cytoplasm, which is everything inside the cell with the exception of the nucleus, inside. So that's all the fluid as well as the organelles. Okay, that's the cytoplasm. Cytosol, right there, cytosol is just the fluid inside the cell, which has some different characteristics. There's different things inside the cytosol, different things that would change its viscosity and other things like that. All right, so now as we're describing cells, we have what's called extracellular fluid, which means outside. And then we have intracellular fluid, which is inside. Another name for extracellular fluid, which is outside, is interstitial fluid. Interstitial fluid, okay? And we need to know that so we have an idea when I say things like extracellular, that you know that I'm talking outside, or interstitial is in between the cells but outside the cell also. Okay. So the first item, organelle, we want to describe here is we want to describe the centrosome or centrioles. That's this guy right here in the center. Okay, and I think I can zoom in a little bit for you guys. This is in the cytoplasm. This is basically kind of considered the hub for the cytoskeleton. So when this cell goes through its mitosis, which we talked about just a second ago, which is cellular reproduction or somatic cell division, this guy splits apart and one goes up north and one goes down south. And then what happens is it sends out microtubules to grab on one side of DNA and one to grow on the other side of the DNA and pulls it apart. And we end up with two perfect copies of DNA in each individual cell, which will show you mitosis here as we get into it a little bit later in this chapter. Okay, all right, so that is the centrosome with the two centrioles on the inside. Okay, all right, next we have the cytoskeleton, which we talked about before. So what does a skeleton do for us as humans? Well, it gives us structure and support, some other things, blood cell production. Well, the cytoskeleton in the cell does that also. It gives it structural support, strength, so that it doesn't break apart. Also, the microtubules can be used as a transport system. So it's like a tube, not unlike the tube at the drive-through at a bank, where you put your car inside the little vacuum thing and it pulls your money or your ID or your check or whatever to the teller across the way. So the same thing happens as the microtubules that actually allows it to transport across the cell. All right, so there's the functions: function, support, movement of cellular structures and materials. Okay. All right, so let's move. So I'm trying to bring that up a little bit, but there we go. All right, next is the plasma membrane. Now, in the last chapter, in the chemistry section, we talked about the phospholipids. Phospholipid bilayer is what makes up the plasma membrane or cellular membrane on the outside. Okay, it is a two layers of the phospholipids, and it has things like these integral proteins, which are integral just means part of, and this is how we get large molecules to pass through these integral proteins. But otherwise, the plasma membrane keeps this bad stuff out of the cell and the good stuff inside the cell, and it only allows things to come in the cell that need to be coming in, and it allows the things leaving the cell to leave. Right, so you can think of metabolism, right? So metabolism is all chemical reactions in the body at any given moment. That's the definition. So the process of metabolism creates metabolites. All right, those are byproducts of that chemical reaction. And of course, metabolites have to leave the cell, return to the interstitial fluid, then do what? Then they have to be able to get, let me manage some participants here if someone wants to come in. Okay. All right, so some of these metabolites have to come in and some have to go out. That's all chemical reaction stuff. Okay. All right. So that is, and we'll look more at the plasma membrane here in a little bit. So you can see how it isolation, protection, and that stuff. Okay.
You can't even rename
it. Next we have the cilia.
We have to change it.
The cilia. All right, so cilia are long extensions of, well, we actually have two. So if you look at here at the top of the brush border, all right, we have some of the microvilli and the cilia. The cilia are long hair-like projections that actually move and they beat and allow stuff to move across the surface of the cell. So the cilia is movement, not necessarily of the cell, but movement of stuff across the surface. For example, as you sit next to people around you, as you're sitting in classrooms, things like that, this your partner, your neighbor is sloughing skin, and that skin is then going into your mouth and your nose, and you inhale it, and then it actually goes into your lung areas, and then it gets picked up by the mucus in there. Now all that skin slough, dust, all that gets attached or in the mucus, and then the cilia will actually move it out with a beat. So they all kind of wave together in a beat to move stuff across the surface to leave so that it can expel it out of your lungs, which then at that point you'll either hack it up or swallow it. Most of the time you just swallow it. So tell your neighbors to quit sloughing their skin on you. Next, we have microvilli, which is what they're showing here on top. And the microvilli, you can imagine that using the microvilli will increase the surface area of the cell here. So where you think logically, where would you need an increase in surface area of a cell? Well, that would be in your gut for digestion or absorption. So we increase the surface area to be more efficient at retrieving nutrients from the from our digestive materials. Okay. All right. So now we have, so that's the cilia, that's the microvilli. We have proteasomes. Now proteasomes are proteinosomes. That's how I like to call them, as proteinosomes. But they are proteasomes. They're hollow cylinders that actually have on the end some specific receptor kind of proteins that actually have a shape to them. And as we talked before, in proteins in the lab, the most important thing about proteins is its shape. And that's where we talked about the primary, secondary, tertiary, and quaternary. Well, this very particular shape will allow it to attach to certain proteins inside the cell, where then this proteasomes will actually break down cellular damaged or abnormal proteins. Okay, and of course you'll look at abnormal intracellular proteins. And this, what does intracellular mean? It's inside the cell. Okay. Next we have ribosomes, and there's two types of the ribosomes. First of all, they're just protein synthesizers. What they do is actually the DNA creates mRNA, which is called messenger RNA. It sends that messenger RNA outside the nuclear envelope through nuclear pores and goes into the rough ER, but attaches to the ribosomes, the fixed ribosomes. We just said they're protein synthesizers, so that messenger RNA will then create a protein. Now that's the fixed one, will make proteins to be shipped out of the cell to go do a job. Whereas, of course, inside the cell, we need proteins in there as well. So we have these free ribosomes, which you see cruising around through the cytoplasm. Remember, cytoplasm is everything inside the cell but the nucleus. So then these guys will make proteins from these guys floating for this cell. So the free ribosomes are for protein synthesis inside this cell. Okay. And we'll talk more about that. Transcription, translation is what that's called, by the way, of the proteins at a later time. All right, next we have peroxisomes. Now peroxisomes, it's showing you this vesicle. It's a purple vesicle, and vesicles are nothing more than just pouches that contain something. So if you hear the word vesicle, it's just a pouch. Okay, it's just a pouch. So peroxisomes have oxidative enzymes that break down things from fatty acid metabolism, will create things that need to be broken down that are oxidative. So we have oxidative enzymes inside the peroxisomes. Right? Then we have lysosomes, which are digestive enzymes. Those digestive enzymes are shown you here in the green vesicles. They will destroy anything in here, like a worn out organelle. Some cells can actually phagocytize, in other words, eat pathogens. Right? So this cell could come up to a bacteria and open wide and bring it in, and then what happens is the vesicles will then attach to that, release their digestive enzymes, and destroy that bacterium or whatever that might be. Okay, so digestive enzymes are just breaking down things that are not supposed to be there or are damaged. Okay, so that is lysosomes. Now, lysosomes actually will they're created from this thing, which is the next thing, which is the Golgi apparatus. Now, Golgi apparatus is typically what we call FedEx, or I call FedEx. It's a packaging and shipping organelle. So from the rough ER, a vesicle will transmit or move, how's that? Move to the Golgi apparatus, where we'll attach there and then release these newly synthesized proteins from the ribosomes. The rough ER synthesizes and packages and changes it a little bit, puts it in a vesicle, and then sends it to Golgi apparatus. Remember, these fixed ribosomes create proteins for outside the cell. So then it sends it over to Golgi, FedEx, which packages it up and sends it outside the cell through secreting vesicles, pouches, right? One of the things, like I was alluding to, is the lysosomes are also created at this point. So the lysosomes are released here. They don't leave, but then they come down into this cytoplasm of this cell and perform its job, whatever that might be. Okay, so Golgi apparatus is storage, and storage, packaging, shipping, alteration of proteins. Okay. All right, next you see here we have multiple mitochondria. The mitochondria are the powerhouse of the cell. They generate about 95% of that ATP that we talked about before, which is adenosine triphosphate, which was in chapter 2, and it showed you that picture. And remember those yellow tennis balls. And then where's the energy stored in ATP? It is from those high energy bonds between the two inorganic phosphates. Okay, so when you look at mitochondria, you actually need to describe it in 4 different ways or things. So first of all, it's the powerhouse of the cell, which produces about 95% of the ATP through aerobic respiration. And we'll talk more about that, but some of you guys have already had biology where they've talked about the Krebs cycle, citric acid cycle, tricarboxylic acid cycle. Those are all the same thing. They create ATP, and that's where it happens, is actually inside the cell, which is inside the mitochondria. Okay, this is for aerobic, which means what? With oxygen. All right, so if it's anaerobic respiration, in other words, you're running a marathon and you don't get enough oxygen to your cells, you create a ATP, and only about 5% out here in the cytoplasm or the cytosol of the cell. So if we have oxygen, we get into the mitochondria. So as I was alluding to, the mitochondria has 4 different layers. It has the inner and outer membranes right there, the cristae, which are the folds here, and then the matrix, which is the goo, the liquid inside there. The reason that's important is because it gets into the next step of how ATP is actually created, and it goes through the electron transport chain and things of that nature, which we really don't hit too terribly much in here. Anyway, ATP, okay? That's the mitochondria. Endoplasmic reticulum, or ER, endoplasmic reticulum. So we have the rough ER, which is what we talked about before, and we have the smooth ER. Okay, so the rough ER, this is modifies and packages and ships the newly synthesized proteins, where are they synthesized from? The ribosomes, right, which are attached to the membrane here of this ER, which then comes inside. So that newly packaged and synthesized and modified protein then is shipped out through vesicles to where? Right, to the Golgi apparatus, FedEx. All right, next we have the smooth ER. Now the smooth ER detoxifies chemicals. It also creates lipids. So what might help you with the smooth ER is smooth lips. Smooth lips. So smooth lips, that is the smooth ER. All right, next we have the nucleus, which nucleus means kernel or nut. It is the control center for the cell. There it is right there, of course. Okay, controls the functions of the cell. It has inside of it, it has all of our DNA. The DNA does not leave the nucleus except for when we're going through mitosis. So as you look, you see this dark patches here. Well, these are actually what we call chromatin. Okay, chromatin. And chromatin is uncoiled DNA. And what is our DNA? What does it do for us? It's all our genetic information, right? It tells us to build proteins. It also does our characteristics, hair color, eye color, body habitus, whatever. Okay, that's all genetic information. But protein synthesis is the biggest job. So everything that happens in the cell is protein synthesis, right? Whether that's create mucus or create hormones, more blood, whatever, more plasma, whatever its job is. That's where it gets its information from, is inside the nucleus, and then of course from the DNA. Now, as I said before, when we're actually making proteins and things of that nature, we use DNA to create messenger RNA. DNA to make messenger RNA. We have the complementary base pairs. You remember that C, G, T, A, right? So that actually makes what's called messenger RNA. Okay, looks like a time limit is coming. If this shuts us off at time, I'll just create another one and record that one too. Okay, so just keep your eye out if you want to come back to the next one. Okay. All right, so anyway, this is chromatin. Nucleoplasm is everything inside the nucleus except the nucleolus. Okay, and of course there's some ribosomal subunits in here. Remember what we said, ribosomes, or right, protein synthesis, which actually creates a ribosome. So when you look at the nucleus, we have the nuclear envelope, we have nuclear pores, we have the nucleoplasm, we have the chromatin, and we have the nucleolus. So those are all the things that are inside the nucleus there. Okay, so as you can see, this is a very important spotlight. Okay, so very important spotlight. Now, as we get through the chapter here, you'll see that there are more. They will break these down individually and start talking about these individually, and we'll add a little bit more information, but this is a hugely important spotlight for you guys. That's a huge part of this chapter. Okay. All right. All right, so introduction to cells, we did that kind of already. There's that. Oh, so what did I do that for? Okay, so we can move on to the next. Okay, so 3.1. So this is the plasma membrane or the cell membrane. Remember, we don't have cell walls. We don't have cell walls, that's in plants. Okay. All right, so plasma membrane, it creates physical isolation, regulation exchange with the environment, sensitive to the environment, and structural support, which, yeah, that makes sense. But so here, look, here is your plasma membrane, your phospholipid bilayers. Okay, your phospholipid bilayers. So you have polar heads and non-polar tails, which means we talked about before, which means what? This is hydrophilic, this is hydrophobic, right? Hydrophilic. So what's on the outside of the cell? We talked about it. It is what? Extracellular fluid or interstitial fluid, which is water. And then inside the cell, which is cytosol, which is primarily water. Okay. All right. So then what you see then here, if this is hydrophobic, the tails means it's water fearing, right? Hydrophobic, which means that it's like a lipid or a fat, which makes sense. That why we have then cholesterol. As you look up here, you have cholesterol here. We have cholesterol in here, and that cholesterol actually gives this plasma membrane structural support. Okay, so we have these integral proteins that have passageways in here. We have cytoskeleton inside here. We have these glycoproteins and glycolipids in here, which is our identifier markers, and all this stuff gives this cell membrane structural and support. Okay, structural support. So this is the phospholipid bilayer, polar heads and non-polar tails. Polar heads and non-polar tails. Okay. All right, so here this describes your integral proteins, and your peripheral proteins are bound on the inner and outer surface. So it's peripheral to the membrane, which can be on the inside or the outside. It describes anchoring proteins, so anchoring cells together, the cytoskeleton together. Recognition proteins, these are cell identity markers. This is part of MHC2 proteins. MHC is called major histocompatibility proteins, and these are what give us our, this is our immune system to fight off things that are not us. So this is why this is the markers and the cross and match and all this other stuff that we have to do to protect ourselves for when we have a kidney transplant or a liver transplant. Then these guys, MHC2, are really important. They are what protect us from outside environmental dangers. Okay, so those are recognition proteins. Enzymes, of course, we know that enzymes are proteins, very specific shape for a very specific pathway in some type of some fashion. Receptor proteins, we have these things on the outside called ligands, for example, like glucose on the outside of a cell, and that ligand will come and attach to a very specific receptor on the outside of the cell, which then will allow glucose to come into the cell. And sure, they give the example here. Insulin is also okay. Carrier proteins, channels. Channels are a central pore that forms a passageway through the plasma membrane, and of course we'll talk more about the different types of channels that that allow things in. And when someone says gated channel, it just means there has to be some shape on the receptor that actually is activated when the thing on the outside, the ligand, attaches to it, allows it to bring that stuff in. And of course, we'll talk more about that as we go. Okay. Okay. Membrane carbohydrates or glycocalyx. They're also called the glycocalyx. Proteoglycans, glycoproteins, glycolipids. They have their function here. So the glycocalyx has a variety of important functions, include the following. So the glycocalyx will be the lubrication and protection, anchoring and locomotion, specificity and binding, and recognition. Okay. So now again, guys, as I'm going over those sections, I'm just giving you kind of definition stuff. Make sure you read it so that you understand what that is exactly telling you. Okay. All right, so we said before, as we described when we first started with that spotlight, what the cytoplasm is. So it's material between the plasma membrane and the membrane that surrounds the nucleus. The cytoplasm has three major subdivisions: cytosol, organelles, and inclusions. And inclusions are just bodies inside the cell, like, for example, pigment or fat, little fat bags inside each cell. Okay, cytosol is also known as the intracellular fluid. Remember we talked about intra and extracellular fluids. So intracellular fluid, it's a mixture of water and various dissolved and insoluble materials. Okay, so organelles, there's two different types of organelles. There's membranous and non-membranous organelles. Just pretty straightforward as whether it has a membrane or not. Okay, and of course inclusions are masses of insoluble materials. Some inclusions are stored as nutrients such as glycogen, which glycogen is stored glucose, and we store that either in the skeletal muscle or the liver, right? So glucose in excess will be stored as glycogen. Lipid droplets in fat cells, others pigment granulations as brown skin pigment, melanin found in hair and skin cells. Okay. So what's in the cytosol? Of course, that's the intracellular fluid, or the fluid inside. Okay. So this is showing cytosol different from extracellular fluid. Some important differences between cytosol and extracellular fluid are as follows. Sodium, potassium concentration is different. Now the sodium potassium differentiation here is actually really important. Sodium likes to live outside the cell and potassium likes to live inside the cell. And that difference there is a ability to do work. So when we talked about that potential and kinetic energies, this is one way that, for example, we can get a heart cell to function is because of this difference here, the sodium on the outside and potassium. And remember, we're talking as them as in ions, right? So anions, okay, or cations, okay? So anyway, so so potassium just likes to be on the inside. Sodium likes to be on the outside. Suspended protein concentrations differ. Which makes sense. We have different proteins to do different jobs on the inside and the outside, and nutrient concentrations differ. The cytosol usually contains smaller quantities of carbohydrates, lipids, and smaller levels of amino acids, and does the extracellular fluid. And all that means is it makes sense though, because we don't do any functions outside of the cells. All the chemical reactions occur inside the cells, and the only thing that the fluid is a transport medium. Okay, a transport medium. Okay, so if this thing shuts off in a minute, I will just start up another one. I'll go through and post another link. All right, so it may stop me here in just a second. Okay, let's see how much time we got left. You guys still hear me out there?
Yes.
Okay. All right, so let's see where we're at.
Yes, we